The history of biology traces the study of the living world from ancient to modern times. Although the concept of biology as a single coherent field arose in the Enlightenment century, the biological sciences emerged from traditions of medicine and natural history reaching back to Ayurveda, ancient Egyptian medicine and the works of Aristotle and Galen in the ancient Greco-Roman world. This ancient work was further developed in the Middle Ages by Muslim physicians and scholars such as Avicenna. During the European Renaissance and early modern period, biological thought was revolutionized in Europe by a renewed interest in empiricism and the discovery of many novel organisms. Prominent in this movement were Vesalius and Harvey, who used experimentation and careful observation in physiology, and naturalists such as Linnaeus and Buffon who began to classify the diversity of life and the fossil record, as well as the development and behavior of organisms. Antony van Leeuwenhoek revealed by means of microscopy the previously unknown world of microorganisms, laying the groundwork for cell theory. The growing importance of natural theology, partly a response to the rise of mechanical philosophy, encouraged the growth of natural history although it entrenched the argument from design. The frontispiece to Erasmus Darwin's evolution-themed poem The Temple of Nature shows a goddess pulling back the veil from nature in the person of Artemis. Allegory and metaphor have often played an important role in the history of biology. Over the Uttradharma and Enlightenment centuries, biological sciences such as botany and zoology became increasingly professional scientific disciplines. Lavoisier and other physical scientists began to connect the animate and inanimate worlds through physics and chemistry. Explorer naturalists such as Alexander von Humboldt investigated the interaction between organisms and their environment, and the ways this relationship depends on geography, laying the foundations for biogeography, ecology and ethology. Naturalists began to reject essentialism and consider the importance of extinction and the mutability of species. Cell theory provided a new perspective on the fundamental basis of life. These developments, as well as the results from embryology and paleontology, were synthesized in Charles Darwin's theory of evolution by natural selection. The end of the Enlightenment century saw the fall of spontaneous generation and the rise of the germ theory of disease, though the mechanism of inheritance remained a mystery. In the early 20th century, the rediscovery of Mendel's work led to the rapid development of genetics by Thomas Hunt Morgan and his students, and by the 18th suffered the combination of population genetics and natural selection in the new Darwinian synthesis. New disciplines developed rapidly, especially after Watson and Crick proposed the structure of DNA. Following the establishment of the central dogma and the cracking of the genetic code, biology was largely split between organismal biology, the fields that deal with whole organisms and groups of organisms, and the fields related to cellular and molecular biology. By the late 20th century, new fields like genomics and proteomics were reversing this trend, with organismal biologists using molecular techniques, and molecular and cell biologists investigating the interplay between genes and the environment, as well as the genetics of natural populations of organisms. The earliest humans must have had and passed on knowledge about plants and animals to increase their chances of survival. This may have included knowledge of human and animal anatomy and aspects of animal behavior, such as migration patterns. However, the first major turning point in biological knowledge came with the Neolithic revolution about 10,000 years ago. Humans first domesticated plants for farming, then livestock animals to accompany the resulting sedentary societies. 1. In around 3000 to 1200 BCE, the ancient Egyptians and Mesopotamians made contributions to astronomy, mathematics, and medicine, which later entered and shaped Greek natural philosophy of classical antiquity, a period that profoundly influenced the development of what came to be known as biology. Ancient Egypt. Over a dozen medical papyri have been preserved, most notably the Edwin Smith Papyrus, the oldest extant surgical handbook, and the Ebers Papyrus, a handbook of preparing and using materia medica for various diseases, both from around 800 BCE. Ancient Egypt is also known for developing embalming, which was used for mummification, in order to preserve human remains and forestall decomposition. Mesopotamia. Further information, Babylonian medicine. The Mesopotamians seem to have had little interest in the natural world as such, preferring to study how the gods had ordered the universe. Animal physiology was studied for divination, including especially the anatomy of the liver, seen as an important organ in haruspicy. Animal behavior too was studied for divinatory purposes. Most information about the training and domestication of animals was probably transmitted orally, but one text dealing with the training of horses has survived. The ancient Mesopotamians had no distinction between rational science and magic. When a person became ill, doctors prescribed both magical formulas to be recited and medicinal treatments. The earliest medical prescriptions appear in Sumerian during the 3rd dynasty of c. 2112 c. 2004 BCE. The most extensive Babylonian medical text, however, is the diagnostic handbook written by the Ummanu, or chief scholar, Esagil Kinapli of Bossipa, during the reign of the Babylonian king Adad-apla Iddina 104 se 104 BCE. In East Semitic cultures, the main medicinal authority was an exorcist healer known as an asipu. The profession was passed down from father to son and was held in high regard. Of less frequent recourse was the asu, a healer who treated physical symptoms using remedies composed of herbs, animal products, and minerals, as well as potions, enemas, and ointments or poultices. These physicians, who could be either male or female, also dressed wounds, set limbs, and performed simple surgeries. The ancient Mesopotamians also practiced prophylaxis and took measures to prevent the spread of disease. The European Renaissance brought expanded interest in both empirical natural history and physiology. In 1543, Andreas Vesalius inaugurated the modern era of Western medicine with his seminal human anatomy treatise De Humani Corporis Fabrica, which was based on dissection of corpses. Vesalius was the first in a series of anatomists who gradually replaced scholasticism with empiricism in physiology and medicine, relying on first-hand experience rather than authority and abstract reasoning. Via herbalism, medicine was also indirectly the source of renewed empiricism in the study of plants. Otto Brunfels, Hieronymus Bock and Leonhard Fuchs wrote extensively on wild plants, the beginning of a nature-based approach to the full range of plant life. Bestiaries, a genre that combines both the natural and figurative knowledge of animals, also became more sophisticated, especially with the work of William Turner, Pierre Bellon, Guillaume Rondelet, Conrad Gessner, and Ulysse Aldrovandi. Artists such as Albrecht Dürer and Leonardo da Vinci, often working with naturalists, were also interested in the bodies of animals and humans, studying physiology in detail and contributing to the growth of anatomical knowledge. The traditions of alchemy and natural magic, especially in the work of Paracelsus, also laid claim to knowledge of the living world. Alchemists subjected organic matter to chemical analysis and experimented liberally with both biological and mineral pharmacology. This was part of a larger transition in worldviews, the rise of the mechanical philosophy, that continued into the 17th century, as the traditional metaphor of nature as organism was replaced by the nature as machine metaphor. The pre-Socratic philosophers asked many questions about life but produced little systematic knowledge of specifically biological interest, though the attempts of the atomists to explain life in purely physical terms would recur periodically through the history of biology. However, the medical theories of Hippocrates and his followers, especially humorism, had a lasting impact. The philosopher Aristotle was the most influential scholar of the living world from classical antiquity. Though his early work in natural philosophy was speculative, Aristotle's later biological writings were more empirical, focusing on biological causation and the diversity of life. He made countless observations of nature, especially the habits and attributes of plants and animals in the world around him, which he devoted considerable attention to categorizing. In all, Aristotle classified 540 animal species and dissected at least 50. He believed that intellectual purposes, formal causes, guided all natural processes. Aristotle, and nearly all Western scholars after him until the 18th century, believed that creatures were arranged in a graded scale of perfection rising from plants on up to humans, the Scala Naturae or great chain of being. Aristotle's successor at the Lyceum, Theophrastus, wrote a series of books on botany, the history of plants, which survived as the most important contribution of antiquity to botany, even into the Middle Ages. Many of Theophrastus' names survive into modern times, such as Carpos for fruit and Pericarpion for seed vessel. Dioscorides wrote a pioneering and encyclopedic pharmacopoeia, the Materia Medica, incorporating descriptions of some 600 plants and their uses in medicine. Pliny the Elder, in his Natural History, assembled a similarly encyclopedic account of things in nature, including accounts of many plants and animals. A few scholars in the Hellenistic period under the Ptolemies, particularly Herophilus of Chalcedon and Erasistratus of Chios, amended Aristotle's physiological work, even performing dissections and vivisections. Claudius Galen became the most important authority on medicine and anatomy. Though a few ancient atomists such as Lucretius challenged the teleological Aristotelian viewpoint that all aspects of life are the result of design or purpose, teleology and after the rise of Christianity, natural theology would remain central to biological thought essentially until the 18th and 19th centuries. Ernst W. Mayr argued that nothing of any real consequence happened in biology after Lucretius and Galen until the Renaissance. The ideas of the Greek traditions of natural history and medicine survived, but they were generally taken unquestioningly in medieval Europe. The decline of the Roman Empire led to the disappearance or destruction of much knowledge, though physicians still incorporated many aspects of the Greek tradition into training and practice. In Byzantium and the Islamic world, many of the Greek works were translated into Arabic and many of the works of Aristotle were preserved. The Art Venendi, by Frederick II, Holy Roman Emperor, was an influential medieval natural history text that explored bird morphology. During the High Middle Ages, a few European scholars such as Hildegard of Bingen, Albertus Magnus and Frederick II wrote on natural history. The rise of European universities, though important for the development of physics and philosophy, had little impact on biological scholarship. Observations and theories regarding nature and human health, separate from Western traditions, had emerged independently in other civilizations such as those in China and the Indian subcontinent. In ancient China, earlier conceptions can be found dispersed across several different disciplines, including the work of herbologists, physicians, alchemists, and philosophers. The Taoist tradition of Chinese alchemy, for example, emphasized health with the ultimate goal being the elixir of life. The system of classical Chinese medicine usually revolved around the theory of yin and yang, and the five phases. Taoist philosophers, such as Zhuangzi in the 4th century BCE, also expressed ideas related to evolution, such as denying the fixity of biological species and speculating that species had developed different attributes in response to different environments. One of the oldest organized systems of medicine is known from ancient India in the form of Ayurveda, which originated around 1500 BCE from Atharva Veda, one of the four most ancient books of Indian knowledge, wisdom and culture. The ancient Indian Ayurveda tradition independently developed the concept of three humours, resembling that of the four humours of ancient Greek medicine, though the Ayurvedic system included further complications, such as the body being composed of 5 elements and 7 basic tissues. Ayurvedic writers also classified living things into 4 categories based on the method of birth, from the womb, eggs, heat or moisture, and seeds, and explained the conception of a fetus in detail. They also made considerable advances in the field of surgery, often without the use of human dissection or animal vivisection. One of the earliest Ayurvedic treatises was the Sushruta Samhita, attributed to Sushruta in the 6th century BCE. It was also an early Materia Medica, describing 700 medicinal plants, 64 preparations from mineral sources, and 57 preparations based on animal sources. Systematizing, naming and classifying dominated natural history throughout much of the 17th and 18th centuries. Carl Linnaeus published a basic taxonomy for the natural world in 1735, variations of which have been in use ever since, and in the 1750s introduced scientific names for all his species. While Linnaeus conceived of species as unchanging parts of a designed hierarchy, the other great naturalist of the 18th century, Georges-Louis Leclerc, Comte de Buffon, treated species as artificial categories and living forms as malleable, even suggesting the possibility of common descent. Though he was opposed to evolution, Buffon is a key figure in the history of evolutionary thought. His work would influence the evolutionary theories of both Lamarck and Darwin. The discovery and description of new species and the collection of specimens became a passion of scientific gentlemen and a lucrative enterprise for entrepreneurs. Many naturalists traveled the globe in search of scientific knowledge and adventure. Cabinets of curiosities, such as that of Olevum, were centers of biological knowledge in the early modern period, bringing organisms from across the world together in one place. Before the age of exploration, naturalists had little idea of the sheer scale of biological diversity. Extending the work of Vesalius into experiments on still living bodies, of both humans and animals, William Harvey and other natural philosophers investigated the roles of blood, veins and arteries. Harvey's De Motu Cordis in 1628 was the beginning of the end for Galenic theory, and alongside Santorio's studies of metabolism, it served as an influential model of quantitative approaches to physiology. In the early 17th century, the micro-world of biology was just beginning to open up. A few lensmakers and natural philosophers had been creating crude microscopes since the late 16th century, and Robert Hooke published the seminal Micrographia based on observations with his own compound microscope in 1665. But it was not until Antony van Leeuwenhoek's dramatic improvements in lensmaking beginning in the 1670s, ultimately producing up to 200-fold magnification with a single lens, that scholars discovered spermatozoa, bacteria, infusoria and the sheer strangeness and diversity of microscopic life. Similar investigations by Jan Swammerdam led to a new interest in entomology and built the basic techniques of microscopic dissection and staining. In Micrographia, Robert Hooke had applied the word cell to biological structures such as this piece of cork, but it was not until the 19th century that scientists considered cells the universal basis of life. As the microscopic world was expanding, the macroscopic world was shrinking. Naming and classifying such as natural history throughout much of the 17th and 18th centuries. From across the globe, Linnaeus published a basic taxonomy for the natural theology of 1735, variations of which have been in use ever since the Nietzschean catalyzed the development of paleontology. Names for 1669, Nicholas Steno published an essay on how the remaining parts of organisms could be trapped in layers of sediment naturalist of the 18th century. Although Steno's ideas about fossilization were species as artificial categories and philosophers as malleable, an origin for all fossils would not be accepted by all naturalists until the end of the 18th century. Buffon is a key figure in the history of evolutionary thought, such as his age of influence and the evolutionary theories of both Lamarck and Darwin. The discovery and description of new species and the collection of specimens became a passion of scientific gentlemen and a lucrative enterprise for entrepreneurs. Many naturalists traveled the globe in search of scientific knowledge and adventure. Cabinets of curiosities, such as that of Olevam, were centers of biological knowledge in the early modern period, bringing organisms from across the world together in one place. Before the age of exploration, naturalists had little idea of the sheer scale of biological diversity. Extending the work of Vesalius into experiments on still living bodies of both humans and animals, William Harvey and other natural philosophers investigated the roles of blood, veins and arteries. Harvey's De Motu Cordis in 1628 was the beginning of the end for Galenic theory, and alongside Santorio's studies of metabolism, it served as an influential model of quantitative approaches to physiology. In the early 17th century, the micro-world of biology was just beginning to open up. A few lensmakers and natural philosophers had been creating crude microscopes since the late 16th century, and Robert Hooke published the seminal Micrographia based on observations with his own compound microscope in 1665. But it was not until Antony van Leeuwenhoek's dramatic improvements in lensmaking beginning in the 1670s, ultimately producing up to 200-fold magnification with a single lens, that scholars discovered spermatozoa, bacteria, infusoria and the sheer strangeness and diversity of microscopic life. Similar investigations by Jan Swammerdam led to a new interest in entomology and built the basic techniques of microscopic dissection and staining. In Micrographia, Robert Hooke had applied the word cell to biological structures such as this piece of cork, but it was not until the 19th century that scientists considered cells the universal basis of life. As the microscopic world was expanding, the macroscopic world was shrinking. Botanists such as John Ray worked to incorporate the flood of newly discovered organisms shipped from across the globe into a coherent taxonomy and a coherent theology, natural theology. Debate over another flood, the Nochian, catalyzed the development of paleontology. In 1669, Nicholas Steno published an essay on how the remains of living organisms could be trapped in layers of sediment and mineralized to produce fossils. Although Steno's ideas about fossilization were well known and much debated among natural philosophers, an organic origin for all fossils would not be accepted by all naturalists until the end of the 18th century due to philosophical and theological debate about issues such as the age of the earth and extinction. The most significant evolutionary theory before Darwin's was that of Jean-Baptiste Lamarck, based on the inheritance of acquired characteristics, an inheritance mechanism that was widely accepted until the 20th century. It described a chain of development stretching from the lowliest microbe to humans. The British naturalist Charles Darwin, combining the biogeographical approach of Humboldt, the uniformitarian geology of Lyell, Thomas Malthus's writings on population growth, and his own morphological expertise, created a more successful evolutionary theory based on natural selection. Similar evidence led Alfred Russel Wallace to independently reach the same conclusions. The 1859 publication of Darwin's theory On the Origin of Species by Means of Natural Selection, or the preservation of favored races in the struggle for life, is often considered the central event in the history of modern biology. Darwin's established credibility as a naturalist, the sober tone of the work, and most of all the sheer strength and volume of evidence presented, allowed Origin to succeed where previous evolutionary works such as the anonymous vestiges of creation had failed. Most scientists were convinced of evolution and common descent by the end of the 19th century. However, natural selection would not be accepted as the primary mechanism of evolution until well into the 20th century, as most contemporary theories of heredity seemed incompatible with the inheritance of random variation. Charles Darwin's first sketch of an evolutionary tree from his first notebook on transmutation of species 1837. Wallace, following on earlier work by de Candolle, Humboldt and Darwin, made major contributions to zoogeography. Because of his interest in the transmutation hypothesis, he paid particular attention to the geographical distribution of closely allied species during his fieldwork first in South America and then in the Malay archipelago. While in the archipelago he identified the Wallace Line, which runs through the Spice Islands dividing the fauna of the archipelago between an Asian zone and a New Guinea, Australian zone. His key question, as to why the fauna of islands with such similar climates should be so different, could only be answered by considering their origin. In 1876 he wrote The Geographical Distribution of Animals, which was the standard reference work for over half a century, and a sequel, Island Life, in 1880 that focused on island biogeography. He extended the six-zone system developed by Philip Sclater for describing the geographical distribution of birds to animals of all kinds. His method of tabulating data on animal groups in geographic zones highlighted the discontinuities, and his appreciation of evolution allowed him to propose rational explanations, which had not been done before. The scientific study of heredity grew rapidly in the wake of Darwin's Origin of Species with the work of Francis Galton and the biometricians. The origin of genetics is usually traced to the 1866 work of the monk Gregor Mendel, who would later be credited with the laws of inheritance. However, his work was not recognized as significant until 35 years afterward. In the meantime, a variety of theories of inheritance based on pangenesis, orthogenesis, or other mechanisms were debated and investigated vigorously. Embryology and ecology also became central biological fields, especially as linked to evolution and popularized in the work of Ernst Haeckel. Most of the 19th century work on heredity, however, was not in the realm of natural history, but that of experimental physiology. 1900 marked the so-called rediscovery of Mendel. Hugo de Vries, Carl Correns, and Erich von Schimmick independently arrived at Mendel's laws which were not actually present in Mendel's work. Soon after, cytologists, cell biologists proposed that chromosomes were the hereditary material. Between 1910 and 1915, Thomas Hunt Morgan and the Drosophilists, in his Fly lab forged these two ideas, both controversial, into the Mendelian chromosome theory of heredity. They quantified the phenomenon of genetic linkage and postulated that genes reside on chromosomes like beads on string. They hypothesized crossing over to explain linkage and constructed genetic maps of the fruit fly Drosophila melanogaster, which became a widely used model organism. Hugo de Vries tried to link the new genetics with evolution, building on his work with heredity and hybridization. He proposed a theory of mutationism, which was widely accepted in the early 20th century. Lamarckism, or the theory of inheritance of acquired characteristics, also had many adherents. Darwinism was seen as incompatible with the continuously variable traits studied by biometricians, which seemed only partially heritable. In the 1920s and 1930s, following the acceptance of the Mendelian chromosome theory, the emergence of the discipline of population genetics, with the work of R.A. Fisher, J.B.S. Haldane and Sewell Wright, unified the idea of evolution by natural selection with Mendelian genetics, producing the modern synthesis. The inheritance of acquired characters was rejected, while mutationism gave way as genetic theories matured. In the second half of the century, the ideas of population genetics began to be applied in the new discipline of the genetics of behavior, sociobiology, and, especially in humans, evolutionary psychology. In the 1960s, W.D. Hamilton and others developed game theory approaches to explain altruism from an evolutionary perspective through kin selection. The possible origin of higher organisms through endosymbiosis, and contrasting approaches to molecular evolution in the gene-centered view, which held selection as the predominant cause of evolution, and the neutral theory, which made genetic drift a key factor, spawned perennial debates over the proper balance of adaptationism and contingency in evolutionary theory. In the 1970s, Stephen Jay Gould and Niles Eldredge proposed the theory of punctuated equilibrium, which holds that stasis is the most prominent feature of the fossil record, and that most evolutionary changes occur rapidly over relatively short periods of time. In 1980, Luis Alvarez and Walter Alvarez proposed the hypothesis that an impact event was responsible for the Cretaceous-Paleogene extinction event. Also in the early 1980s, statistical analysis of the fossil record of marine organisms published by Jacques Sepkoski and David M. Raup led to a better appreciation of the importance of mass extinction events to the history of life on Earth.
By the end of the 19th century, all of the major pathways of drug metabolism had been discovered, along with the outlines of protein and fatty acid metabolism and urea synthesis. In the early decades of the 20th century, the minor components of foods in human nutrition, the vitamins, began to be isolated and synthesized. Improved laboratory techniques such as chromatography and electrophoresis led to rapid advances in physiological chemistry, which, as biochemistry, began to achieve independence from its medical origins. In the 1920s and 1930s, biochemists, led by Hans Krebs and Karl and Gerty Cori, began to work out many of the central metabolic pathways of life, the citric acid cycle, glycogenesis and glycolysis, and the synthesis of steroids and porphyrins. Between the 1930s and 1950s, Fritz Lippmann and others established the role of ATP as the universal carrier of energy in the cell, and mitochondria as the powerhouse of the cell. Such traditionally biochemical work continued to be very actively pursued throughout the 20th century and into the 21st. Origins of molecular biology. Following the rise of classical genetics, many biologists, including a new wave of physical scientists in biology, pursued the question of the gene and its physical nature. Warren Weaver, head of the science division of the Rockefeller Foundation, issued grants to promote research that applied the methods of physics and chemistry to basic biological problems, coining the term molecular biology for this approach in 1938. Many of the significant biological breakthroughs of the 1930s and 1940s were funded by the Rockefeller Foundation. Wendell Stanley's crystallization of tobacco mosaic virus as a pure nucleoprotein in 1935 convinced many scientists that heredity might be explained purely through physics and chemistry. Like biochemistry, the overlapping disciplines of bacteriology and virology, later combined as microbiology, situated between science and medicine, developed rapidly in the early 20th century. Felix Dierel's isolation of bacteriophage during World War I initiated a long line of research focused on phage viruses and the bacteria they infect. The development of standard, genetically uniform organisms that could produce repeatable experimental results was essential for the development of molecular genetics. After early work with Drosophila and maize, the adoption of simpler model systems like the bread mold Neurospora crassa made it possible to connect genetics to biochemistry. Most importantly with Beadle and Tatum's one gene one enzyme hypothesis in 1941. Genetics experiments on even simpler systems like tobacco mosaic virus and bacteriophage, aided by the new technologies of electron microscopy and ultracentrifugation, forced scientists to re-evaluate the literal meaning of life, virus heredity and reproducing nucleoprotein cell structures outside the nucleus. Plasmogens complicated the accepted Mendelian chromosome theory. The central dogma of molecular biology, originally a dogma only in jest, was proposed by Francis Crick in 1958. This is Crick's reconstruction of how he conceived of the central dogma at the time. The solid lines represent, as it seemed in 1958, known modes of information transfer, and the dashed lines represent postulated ones. Oswald Avery showed in 1942 that DNA was likely the genetic material of the chromosome, not its protein. The issue was settled decisively with the 1952 Hershey-Chase experiment, one of many contributions from the so-called phage group centered around physicist and biologist Max Delbruck.
Biotechnology in the general sense has been an important part of biology since the late 19th century. With the industrialization of brewing and agriculture, chemists and biologists became aware of the great potential of human-controlled biological processes. In particular, fermentation proved a great boon to chemical industries. By the early 1970s, a wide range of biotechnologies were being developed, from drugs like penicillin and steroids to foods like chlorella and single-cell protein to gasohol, as well as a wide range of hybrid high-yield crops and agricultural technologies, the basis for the Green Revolution. Carefully engineered strains of the bacterium Escherichia coli are crucial tools in biotechnology as well as many other biological fields. Recombinant DNA. Biotechnology in the modern sense of genetic engineering began in the 1970s, with the invention of recombinant DNA techniques. Restriction enzymes were discovered and characterized in the late 1960s, following on the heels of the isolation, then duplication, then synthesis of viral genes. Beginning with the lab of Paul Berg in 1972, aided by E. Corey from Herbert Boyer's lab, building on work with ligase by Arthur Kornberg's lab, molecular biologists put these pieces together to produce the first transgenic organisms. Soon after, others began using plasmid vectors and adding genes for antibiotic resistance, greatly increasing the reach of the recombinant techniques. Wary of the potential dangers, particularly the possibility of a prolific bacteria with a viral cancer-causing gene, the scientific community as well as a wide range of scientific outsiders reacted to these developments with both enthusiasm and fearful restraint. Prominent molecular biologists led by Berg suggested a temporary moratorium on recombinant DNA research until the dangers could be assessed and policies could be created. This moratorium was largely respected, until the participants in the 1975 Asilomar Conference on Recombinant DNA created policy recommendations and concluded that the technology could be used safely. Following Asilomar, new genetic engineering techniques and applications developed rapidly. DNA sequencing methods improved greatly, pioneered by Frederick Sanger and Walter Gilbert, as did oligonucleotide synthesis and transfection techniques. Researchers learned to control the expression of transgenes, and were soon racing, in both academic and industrial contexts, to create organisms capable of expressing human genes for the production of human hormones. However, this was a more daunting task than molecular biologists had expected. Developments between 1977 and 1980s showed that, due to the phenomena of split genes and splicing, higher organisms had a much more complex system of gene expression than the bacterial models of earlier studies. The first such race, for synthesizing human insulin, was won by Genentech. This marked the beginning of the biotech boom, and with it, the era of gene patents with an unprecedented level of overlap between biology, industry, and law. Molecular systematics and genomics. Further information, history of molecular evolution. By the protein sequencing had already transformed methods of scientific classification of organisms, especially cladistics, but biologists soon began to use RNA and DNA sequences as characters. This expanded the significance of molecular evolution within evolutionary biology, as the results of molecular systematics could be compared with traditional evolutionary trees based on morphology. At the beginning of the 21st century, biological sciences converged with previously differentiated new and classic disciplines like physics into research fields like biophysics. Advances were made in analytical chemistry and physics instrumentation, including improved sensors, optics, traces, instrumentation, signal processing, networks, robots, satellites, and compute power for data collection, storage, analysis, modeling, visualization, and simulations. These technological advances allowed theoretical and experimental research, including internet publication of molecular biochemistry, biological systems, and ecosystem science. This enabled worldwide access to better measurements, theoretical models, complex simulations, theory predictive model experimentation, analysis, worldwide internet observational data reporting, open peer review, collaboration, and internet publication. New fields of biological sciences research emerged, including bioinformatics, neuroscience, theoretical biology, computational genomics, astrobiology, and synthetic biology.